A lot of people have requested that we make a recording available afterward. They can't make the webinar or they'd like to reference it later. So to let you know that the chat box, the Q&A, if you do speak later, it will be recorded and be made available later. So I see a couple of you guys have raised hands. Sit tight. We're gonna get to questions after the formal presentation. And Lisa, is there anything else, housekeeping, we need to cover?
I think you did a great job. Thank you, Sarah.
So with that, I'm going to mute myself and mute my video and turn it over to Lisa. So Lisa, me, and then Jared, and then we'll take questions.
Sounds great. Thanks, Sarah.
Thank you.
Good morning, everyone. Live from Asheville, Friday morning. We are really doing well at TACF. I just wanted to use this opportunity to thank all of our staff and our volunteer leaders who have really stepped up during this uncertain time. We had a lot of realignment and restructuring to do very quickly. Speaking to you, of course, from my home in Weaverville, north of Asheville. Like so many of us have transitioned to work from home. The Asheville staff is pretty much all working from home. They've been incredibly flexible, cheerful, and can do. I'm so proud of all of our staff. And we do have our wonderful Shayna going in twice a week and working with Judy to be sure that we have our mail monitored. Our gifts are still coming in. Thank you so much. During this uncertain time, it's very inspiring. People are still joining and giving to us. And thanks to the foresight and the generosity of so many before us, we are fiscally pretty stable. We're obviously tightening our belts. We've curtailed all travel per the CDC recommendations. And we're following a lot of safety and sanitation protocols, especially on the farm. The farm staff, kudos to them. They are still showing up in Meadowview and doing very careful social distancing and sanitation processes. Dan McKinnon, our farm director, and his staff of Brandon, Jim, Eric, and Lily. They're just a wonderful team and working very hard. Farms don't close down, so they are doing what they can at home, but they also are doing some spacing of their scheduling so they can get the work done on the farm. All of you, I know you'd probably rather be out in the field digging in the dirt, but we've been really busy at home doing a lot of new creative ways of work plans and communicating. Zoom has been very, very helpful. We've been also using Microsoft Teams. Betsy Gamber, our Chief Operating Officer, has been incredible organizing her team to work from home. I wish I could thank every single one of the staff members. There are 17 of us, that's all. So I consider all of our staff members essential at this point because everybody wears a lot of hats and works very, very hard. The other folks I really want to thank this morning are the 7-member executive committee team. It's chaired, of course, by our board chair, Brian McCarthy, and immediate past chair, Michael Duchin, our board secretary and chair of the Promotion and Outreach Committee, Jay Cood, our board treasurer, Barb Tormelin, our senior science advisor, Kim Steiner, and last but not least, our legal counsel, Don Willicke, who is an original founder of the Chestnut Foundation back in 1983. He helped write the charter, and he's still very much involved. And then Steve Barilovitz, of course, our chair of our Science and Tech Committee. I hope you were able to catch the Science and Tech meeting on March 26th. It was highly participatory. We were so happy that the technology worked, and Jared and Sarah, who are our science leadership team, did 2 great presentations. That is recorded. Just let Sarah know if you'd like to see that, and we'll send you a link. So the other stuff going on, we were trying to work very hard in partnership with SUNY College of Environmental Science and Forestry to help them with the open comment period, the public comment period. They have submitted, and it has been accepted, their 280-page, I believe, I know Linda's on here, she could correct me, petition to deregulate the Darling 58 transgenic tree. That's in the hands of USDA APHIS. And we've worked in the last, 3 or 4 months to pull together a task force chaired by Jim Searing, our board member out of Pennsylvania, to really prepare for a positive and high quality and high quantity amounts of positive comments when it does get posted on the Federal Register, which we expect any day now. And speaking of which, I just wanted to have everybody send out good vibes to Andy Newhouse out of ESF. He's taking his final PhD oral exams this morning. So good vibes to you, Andy. You know, we really look forward to getting on the other side of this worrisome time, but I really feel very strongly that we'll come out fine, if not even more strong and more resolute as we go forward. One of the ideas we thought we talk about this morning, and Sarah and Jared can join me in this, is just why we're involved personally, what's important to us as far as the Chestnut Foundation. And when I was called 5 years ago, I just had my 5th year anniversary on, and I'm honored to serve you. But you know, it is a shared leadership model, and I just want to thank all the volunteer leaders out there. But for me personally, I just thought the opportunity to help save an iconic species, that was just too good to be true, and I'm thrilled to be still a part of it. I had a background in land management and philanthropy at the Nature Conservancy, and then I worked at Rollins College for 4 years as a senior administrator. But this has been the best job I've ever loved. It's been really wonderful so far, and mostly because of the people. Obviously, the mission is near and dear to all of our hearts, but I think the people really make this organization sing and shine, and it just keeps me going on a daily basis, the inspiration. I've been trying to reach out to a lot of you personally with phone calls and emails, and I've just gotten so much support and encouragement back, and that just keeps wind in our sails. We really, really appreciate all that you do. I'm trying to think if there's anything else. Oh yeah, the bit of not sad news, but news that we need to share is that we had to make the very tough decision to postpone our in-person meeting in Asheville, North Carolina, October 1st through the 3rd, I believe. And it was a very tough decision. The executive committee was very helpful in helping us make the decision. Part of it was monetary. Part of it is because of the uncertainty of the future of this virus. I know everybody's been watching the news, but some say it's, you know, there's a chance it could reemerge in the fall if it goes down in the summer. So we didn't want to take a chance of not being really careful with our donor money. So we have been working with the hotel to see if we can reschedule for that time next year. So stay tuned for that. And of course, we had to cancel the spring meeting up in Abingdon. So we're going to work really hard to try to connect with you, because we are a family, and it's important that we connect and share stories. So with that, I think I'll turn it over to Sarah with her personal chestnut stories and what's going on in the field as far as the regions. And then Jared will talk a little bit about his story and what's going on at Meadowview Farm, because we've been working very closely with some of those folks. So with that, Sarah, you're ready to roll?
Yeah.
Oh, and we are TACF strong, all right? I know, not particularly original, but we are that. Thanks, everybody.
Hashtags. Thanks, Lisa. So for those of you who don't know me, I'm Sarah Fitzsimmons. I've worked for the American Chestnut Foundation out of our Penn State Partnership Office since 2003. I think I want to hit on a couple things that Lisa did, which is, you know, obviously the mission is important and we're all inspired by the mission. But what I love about my job in particular is being able to work with all the people who are working together to try and restore that iconic species. It's all about expanding the network, and I see a lot of partner participants on here, folks from the Nature Conservancy or Western Pennsylvania Conservancy, colleges and universities obviously play a huge role, but then a lot of the volunteers. The way I got started in the foundation was I was a stand-back intern at the Duke Nicholas School. I was going there in 1999 to 2001, so you guys can do the math. When I was an intern for the Pennsylvania, New Jersey chapter under that program in the summer of 2000. And Paul Sisko, who was the geneticist at the time and the Southern Regional Coordinator, he was the one who helped select me to get that internship position. I slept on Ann and Bob Leffel's futon in their basement in Brogue, Pennsylvania. And for those of you who don't know, Brogue, Pennsylvania isn't the end of the world, but you can see it from there. So I spent the summer there on their farm and traveling around the orchards of Pennsylvania, New Jersey, traveled down to Meadowview, worked a lot there. And when I grew up in Southern West Virginia, and when my papa heard that I was going to be working on this chestnut project, he was the proudest I've ever seen him. He saw the decline of the American chestnut in the mountains of West Virginia from his farm. The mountains that he grew up in, he watched the tree decline. He worked in one-room schoolhouses. He was a teacher, and as people stopped using one-room schoolhouses and those houses or schools started to be torn down, he would salvage all the wood from them. And there was a lot of wormy chestnut that was in those schoolhouses, and he would gather that and put it in his many barns that he has on the farm. And some of that wormy chestnut ended up in the basement of the house that I grew up in. We paneled the basement in that wormy chestnut from one-room schoolhouses. So, you know, I didn't make that connection growing up. You know, I knew that it was wormy chestnut, but I never was inspired to say, oh, I want to save the chestnut when I grow up. But it was just sort of one of those serendipitous connections that were made. So I'm inspired by the story, by the history, by the connection that can be made. And I think I love hearing everybody's stories that run pretty deep in the history of Appalachia and beyond, through these different family connections or just the connection with nature and history. So that's my personal connection. Like I said, I've worked with the foundation full time since 2003. I've been hired at our Penn State Partnership Office, and I oversee our regional and field operations. I work with incredible people. Kendra Collins, who is on this call, she's helping monitor the chat and the Q&A. She works out of our Burlington, Vermont office in coordination with the University of Vermont and the U.S. Forest Service. She oversees our New England operations. I'm at Penn State with Steve Hoy. Hoy is also helping to monitor the chat and the Q&A. And Hoy takes care of the orchards and greenhouse around here at Penn State and throughout Pennsylvania and New Jersey. Tom Sielli is our coordinator for the Mid-Atlantic region. He started out in our southern region, so he's also close to folks down there. But Tom works out of our Charlottesville office and in close alliance with the Virginia Department of Forestry. Right now we have a vacancy at our Southern Regional Science Coordinator. So we're working to try and fill that position. But our regional staff currently, Kendra, Tom, Hoy, myself, we're all working on transitioning from home. We typically travel a great deal, going out to orchards, going out to partners. So it's a bit of a transition to work from home. It is kind of nice. All four of us have young children, all under the age of 10. Tom has 3, I got 2, Hoy's got 2, Kendra's got 1. So we're all trying to juggle that. How do you work from home with 2 full-time parents and children around? So it's been quite an adjustment, but we're working on it. As far as working, I know Hoy and I are both still working some in the field, making sure that we're not losing, well, and Tom and Kendra as well. We want to make sure we don't lose material, that we don't lose traction, that we keep everything alive. So we are able to do absolutely critical activities. But there's obviously going to be some things that have to wait until next year, until 2021, and that's fine. The great thing about trees is they don't go anywhere, I guess. So let's hope they can make it a year for various aspects of what we do. So thank you guys for all that you do. I want to put out a couple of calls to action of what you can do if you're stuck at home. One of our biggest priorities right now is diversification of blight-resistant chestnuts. And the best way to do that is to find wild Americans. If you guys read our magazine, if you read our publications, we've been pushing that for quite a while. We've got a tree locator program. You can download Tree Snap on your phone. If one of your activities that you're allowed to do where you're at is to go on a hike, find trees, use Tree Snap, take a picture of it. Best time to do that would be actually a little bit later in May and June when leaves are actually out. But we need that material, especially Western Tennessee, Northern Alabama, Northern Mississippi, Southern Ohio, Southern Indiana. These are areas that are especially not well represented in our program. So that's one thing you can do. And another thing is just expand the network. Who are other partners that we should work with, and can you encourage other folks to go out there and join us in the work that we do and in our mission. With that, I'm going to turn everything over to Jared and let him share his story and what he's been doing working from home.
Hello, everyone. Welcome to my living room. I'm Jared Westbrook. I'm the Director of Science for the American Chestnut Foundation. I started in 2015, officially, the 2nd of January. And I learned about the job while I was practicing yoga in India, just about to finish my PhD at the University of Florida. My advisor, John Davis, at the University of Florida, sent me this job announcement. And I saw that this job was in Asheville, North Carolina. And I had previously hiked the Appalachian Trail. So I really loved the regions where chestnut grew. And he told me he was on this advisory committee for the looking at the regulatory landscape for the transgenic American chestnut. And one of the things that was very interesting about this regulatory process was, in most cases with transgenic plants, the goal has been to prevent the spread of these genes, foreign genes, into the native populations. Where in this case, with chestnut, we actually do want the genes to spread into the native populations, and we want to use this technology for species restoration. So I thought that was incredibly interesting, that we were using a technology that has been used by big ag, but we're using it now for species restoration. And what does that mean for how we think about how we interact with nature, I guess. And so that really hooked me in. And then I also learned about the breeding program, and that there's been multiple generations of breeding that's been carried out by citizen scientists. And just the staying power of this mission was inspiring. So I'm just incredibly honored to be a part of the mission. So what we're doing now at the farm, we planted like 10,000 seeds before this whole COVID really took off. So we are taking care of those plants right now. We plan on potentially inoculating them if things ease up a little bit in July, although we can keep them alive for a year in the greenhouse. I've been working from home the past month or so, and I'm working on a paper on the small stem assay and looking at how, if we screen seedlings for blight resistance in our breeding program, whether we can get really good, accurate estimates of the resistance of those parent trees from where the seedlings came from. And it turns out that we can. So I'm glad that this is working out. We're also genotyping all of the chapter parents in the breeding program so that we can make better selections. And thus far, we've been able to get about 600 parents from our chapter breeding programs genotyped. We have about 300 more that we have the tissue for, and that is still ongoing at Virginia Tech. The lab technician that is working on preparing those samples is going in when she can. So we plan on continuing with that. So I will send you the data so that we can also make these work smarter in our breeding program. We're still working on the genomes for the Chinese and the American chestnut. Hopefully we have the American chestnut genome published, or where we've already assembled it, and the Chinese genome is well on its way. So we'll be able to compare these genomes and figure out what is the difference between Chinese chestnut and American chestnut that enables the Chinese tree to be resistant and the American to be susceptible. So we're still working where we can. There is going to be a public comment period for the transgenic tree coming up any day now. It's already at the Federal Register. I really think that this is important, that the tree, we have favorable public comments for this tree, and there's basically a number of public comment periods that we'll be going through. This is just the first one with the USDA. Once they review all of the public comments, they draft up an environmental impact statement, and then that also goes out for a second round of public comment. So just looking at that petition, I think it's important for you all that you just talk about why this tree is important, why this mission is important for you. And I think they're going to just see that as, and add a personal touch to it. They're going to see that. And we're also going to have scientists commenting on this as well. It's very important to rigorously evaluate any potential human health or ecological impacts. So scientists will be commenting on those impacts as well. And then we have to go through a public comment period with the EPA as well. So there's numerous chances for you'll be able to interact with this. And so that's what I'm hoping that you guys will positively comment on, so we can have this opportunity to use this technology, which I think is a game changer for us. It really enables us to capture diversity in the breeding program in an easier way, because it's a single gene. If you cross that transgenic pollen onto a wild tree, 50% of the progeny inherit the gene and will be blight tolerant. So it simplifies the process of selection for us tremendously, enabling us to capture a lot more diversity and adaptation. So thank you.
I saw in the chat room, just to follow up on Jared's comments. Thank you, Jared. That was a great summary. Yeah, we'll be sending information and instructions. We've been working on it for weeks and weeks to be sure you all have as easily as possible links and potential comments. They don't want cut and paste. They want discrete, personal, as Jared said, comments to go in. But we will be sending out all kinds of links for you to learn more about the process. If you don't know that much about the transgenic tree and you want to learn more, there's going to be some one-pagers. And of course, the actual petition will be available once it's unembargoed on the ESF website. So we'll send you all those links. There is an executive summary if you don't want to read all 280 pages. So we're going to be absolutely sure that you will get all you need to be able to comment. And if you're not on eSprout, our electronic newsletter, that's a good way to be informed. We do e-blasts. If you aren't on email, or Judy or Shana don't have your email, that's really the way we can communicate right now, especially with this campaign to do positive public commenting. So thank you for bringing that up, Jared, and for your question in the chat room.
Thanks, Lisa, and everybody. So I think what we'll do right now is go to the Q&A. So I know Hoy and Kendra have been collating these questions. Kendra, I think you said your connection has gotten more stable. Do you want to ask us these questions in order?
Yeah, sure.
Okay.
I actually missed the early ordering of things, but I'm just going to go with what's on the list. So the first question from Warren, Sarah, and Jared. Our latest hybrids do not seem to have the shade tolerance we wanted. Is our breeding program going to be changed to get more shade tolerance?
So I'm wondering, how are you measuring shade tolerance? And where is that coming from? Because we haven't noticed that necessarily.
Yeah, I'd have to agree. I don't see any change. We haven't measured it directly. There have been a couple of studies which have shown intermediate shade tolerance of American chestnut, but I don't know that we've studied that directly in the backcross populations extensively. But I haven't seen a decline in performance based just observationally.
Yeah, we have a number of plantings in forest progeny tests, and the plants that are shaded tend to hang out just like a regular American chestnut and be suppressed in the understory. And then when you release them, doing a shelter wood cut or some kind of like opening up a gap, then these trees, they grow incredibly fast. That's they're kind of opportunistic. They just hang out for a long time. And that's how they've been living in the understory since the blight came through. They've just been hanging out and waiting for a gap. And the backcross trees just grow incredibly vigorously after they're released, and the shade and they have a lot of sunlight to grow.
Yeah. All right, Kendra, what's your next?
Can I just add one thing?
Yeah, sure.
I heard from a long-time chestnut grower that the first year they sleep, the second year they creep, and the third year they leap. Have you heard that?
That's kind of for bare root seedlings and hardwood seedlings in general.
Okay.
But yeah, I think chestnuts certainly follow that template as well.
All right, next up. So actually, before questions, I just wanted to note, Linda from ESF shared that the executive summary on their chestnut stuff is on their website, which is in the chat now. So if anyone would like that link, you can check the chat. For our panelists, John Davis asks, do you have materials for people or organizations who might be interested in providing land for nursery or orchard operations?
So I guess I can take that. So we do various types of research plantings for various types of reasons. We have anything from large forest types of installations that we do with partners such as the U.S. Forest Service or like Pennsylvania DCNR. We do occasionally do those with private landowners. And those are usually for silvicultural types of projects. How do we get these trees back into the landscape, or for testing the durability of resistance and American character of our trees? That can be on the order of hundreds to a few thousand trees. We go all the way to our breeding orchards, which are usually about hundreds of trees in size. And those are typically managed a lot more intensively than a lot of people like to plant their trees. We plant them in sort of an orchard type of a style, and usually on a grid, and we request them be maintained for a minimum of 5 years and have data collected and reviewed for 10 to 15 years. And then our current push for diversification of material is for wild type American chestnuts. These have no resistance, but we need to plant them in what we call germplasm conservation orchards. And so we plant those on the order of about 100 trees per location. And then the final smallest category is for the enthusiast. We do make seeds and seedlings available to long-term and sponsor members. And those, we love to hear people's anecdotal observations on those plantings, and we do incorporate them into our overall strategy, but they don't necessarily have stringent scientific guidelines on them. So there are opportunities. Most of them are reviewed on a case-by-case basis. Some of our projects, a lot of people ask about funding because sometimes we do need fencing or lots of other types of supplies to get them in the ground. So some of our projects have taken 5 to 10 years to actually be realized. But it starts at, sort of starting a project, having a vision, and then seeing how we can make that happen.
I should say also, just add to that we've been somewhat limited in the supply of seeds at Meadowview, which is the furthest along in the breeding. We typically harvest somewhere around 50,000 seeds from those orchards. And there's the chapter orchards as well, but a lot of them contain very early stage selection, so they're not the final product yet. We still have some selection of the best parents to do in those programs. And, if we end up going through the regulatory process and getting regulatory approval for transgenic trees, our plan would be to plant orchards, and it would probably take, 7 to 10 years for these trees to start producing seeds. But we would have orchards where we'd have open pollinated, these trees pollinating each other in the open air, and then we could harvest a lot of seeds from those. But again, it's just a matter of finishing our selection and scaling this process up. And it's an exponential process. And right now we're sort of at the beginning stages of that.
All right, Kendra?
Yeah, so John Wenderoff wants to know if we are able to collect and handle emerging leaf tissue for genomic identification this spring. Should we continue sending it to Virginia Tech for processing?
So yes, and you should send it to Meadowview instead. Virginia Tech's labs and mail right now are sort of spotty. I think they're kind of opened, but not And so I wouldn't take a chance of sending leaf tissue to a place where they can't receive it immediately. Whereas we do have our MetaView staff working in a staggered way every day. So I would send it to MetaView instead. And that would be both for, if you have a backcross orchard that we have not collected tissue from, and that you live nearby that orchard and you want to know what the ancestry is of those trees, certainly collect that and make sure you work with your regional coordinator to make sure that you have the trees correctly ID'd and they're already in Dentatabase, because we need to be able to go back and track the pedigrees of those trees when we get the data back. So if you can do that and it's not like you have to travel a long ways, then we still want you to collect from trees that we basically haven't collected from, which are anything that you've selected after 2017, basically. And then also any of these wild trees that you might find, especially in the western part of the American chestnut range. We are still doing genome sequencing on these trees to understand the link between climate adaptation and diversity in the population, so we can develop a strategy for germplasm conservation that maximizes the diversity that we capture and the adaptive capacity of the population. So if you can find these trees, especially in the western part of the range, that's our priority right now. And again, work with your regional coordinator. If you don't know who that is, make sure you contact Tom, Sarah, Kendra right now to figure out what needs to be collected. So they'll work with you. But yes, we are accepting and planning on doing additional genotyping.
And just as an aside, if you are trying to send any material that requires refrigeration or a sample or something, don't send it to universities right now. That includes Penn State. So if you're supposed to send something to me and it needs immediate reception, contact me via email, because you'll need to send it to my home address. All right, Kendra, what's going on?
Well, question for you guys. So we have a bunch of questions all related to transgenic chestnut and the process around that. And then we have a lot of field related questions. So I'm trying to get all the transgenic stuff together. Would you guys like to tackle that first, or do you want to go through some of the field stuff?
Let's go transgenic.
All right. So I apologize to anyone who asked about pruning and planting. We'll get back to that. One sec. So one Drusilla asked, how close are we to getting the transgenic tree approved? And then there's a few questions about comments, but I don't know if you want to address that one first, and then I'll go to the comment questions.
Well, I'll take a shot at that. It's hard to predict. The federal government is obviously very busy right now. Jared, Bill Powell, and I were in Washington, D.C. That was my fourth trip to help cultivate these relationships within the USDA, the U.S. Forest Service, and EPA. They typically take from submission, and Linda on the chat room can correct me here, from submission to actually deregulation, it can be up to 18 months. But once the public comment period opens, once it's on the Federal Register, ready to take any comments, there's 60 days. They have it open for 60 days, and then it's closed, and they do a review, just as Jared said, and issue an environmental impact statement. And they work together with the EPA on that. So just in general, so you all have some hope, because I sure do, the regulators that we've met with, and we got all the way up to an aide to Secretary Perdue in the USDA. We got all the way up to a direct report to Secretary Wheeler in EPA. We've really been working the different levels of these regulation government agencies, and to the person, everyone's been really supportive. What they're hampered by is certain laws that they are chartered to protect and support for human health and so forth. So, there's a lot of excitement within the regulatory agencies for this project, and Bill and Jared did a really great job just last month doing presentations for these folks and showing a real solid a show of unification between our programs and the 3-bird process and how that's going to work to bring back the species. So it's going to be a while, and so we need to stay on it. It's not something we can just do once and hopefully it'll work, but it's gonna take some time. Hope that helped the timing issue.
Yeah, once the public comment period is finished for the USDA, then they take like 6 months to a year to write that environmental impact statement. And then there's another public comment period. And then there's a decision that's made. So you can project this out probably a year or 2 years even from now. And then the EPA process, we're in the process of drafting a petition to them as well. So that hasn't been submitted. So that also has to go through a process, and we don't know how long that will take. So we're still talking probably a year or at least a year, probably more.
All right. So we have a couple on public comments. So John wants to know if we can provide a link or instructions on how to comment. A different John wants to know if we will be sending out an email blast to let rank and file members know when it's time to submit public comments. And Jim wants to know if we can comment on the expected time frame for all the agencies to processes until the transgenic trees can be released, which I think you guys kind of just touched on. So yeah, you want to address our process for comments, public comments?
Sure. Thanks to Sharon at our office and the work of the task force that Jim Searing chairs, we've got a lot of material ready to hit the send button on. We've really tried to get this ready to go. We had heard that this could have been on the Federal Register as early as early March. So we've been furiously getting our messaging together and links where you all can go. And we're going to try to make it as easy as possible for you to participate. So please, if you don't have emails in our system, we'll need your email to be able to communicate with you. We'll probably put some links on social media, like us on our Facebook page, and just keep an eye out on our website as well as ESF's website for ideas and materials, because we've really been gearing up for this for a while. We hired a public relations firm out of DC. We shared the cost of that with ESF, so we're very unified in our desire to get this comment period out and come in a big way, because we do, frankly, anticipate some critics to this. So we want to outshine the critics with our positive comments, especially on the scientific community and citizen scientists. I think those comments, we are told, carry a lot of weight. Hope that answered the question.
And I think on a related, I have a question from Dawn wondering if we can provide the Federal Register date and page where the USDA rule is published. I'm not sure if that exists yet.
It does not. It has not been opened. I know Andy has been in contact with their folks at USDA, and they really can't tell us exactly when, but they say soon or soonish. That's what I've heard. So whatever that means in government time. So just keep an eye on your emails if you can. We'll be sure to send out alerts and let you know how the process is going. We're going to be carefully tracking the comments as they come in. Again, thanks to Sharon and Kristen up in ESF. We're going to be monitoring. We're also trying to get organizations to weigh in on this. That might be a little bit harder to get out of something like the Nature Conservancy, even though I'm an alma mater. But we are, like Jared said, this technology might be a game changer for us in not only tree restoration, but just in conservation in general. So we're working hard, so stay tuned. It's not open yet, but any day now. We'll wake up and all of a sudden the Federal Register will have this on there. So
I just wanted to comment one thing. It's a 60-day comment period. So, I think we'll certainly send out a blast as soon as it opens, but several people have said, will you please remind us? So we'll send reminders, and probably a majority of the comments will likely happen. And we've been told this by many folks who have gone through the process already. Most comments happen at the end of that 60 days. So expect several reminders. If you're not signed up for eSprout, go for it. acf.org, scroll to the bottom of the page, and it says, do you want to sign up for our newsletter? That's how you get eSprout. And that's probably the best way to learn about when that comment period opens.
All right, last question related. I'm going to read the whole thing because it's quite lovely. Anne is joining us from Paris. She says, are you planning social media campaigns to generate wider support for the USDA process, i.e. webinars, Facebook, Twitter, LI, LinkedIn? Not actually sure which one that is. That's how good I am at social media. The ACF story is incredibly compelling. Just wondering how you are thinking of attracting and converting to supporters and donors people who are not yet familiar with ACF. And thank you so very much for this unique opportunity to Zoom and hear from the team, including your personal stories. Based in France, and I'm never able to attend US meetings. Please do more. So happy to see you this morning, Anne, or connect.
Yeah, I mean, I don't know that we've necessarily planned webinars per se, but I think that's a good idea, and especially now that we're doing a lot of things remotely, maybe we should take that, Lisa, and do some education, more build awareness of some more about the process and what we're doing. I know so this Chestnut Chat, we are hoping to turn that into a series. We have some ideas of what we're going to do beyond this. One of the sessions in maybe 2 or 3 weeks that we were hoping to pull together were bring the scientists from SUNY ESF to come and chat, and that might be the perfect opportunity to invite folks to not only learn about the science behind the transgenic chestnut, but also talk a little bit about the public comment period and what people can do to participate in that.
Yeah, well, again, please send your ideas, because we're learning how to do all this stuff virtually when we're so used to being together in person at least a couple of times a year, and being on plantings and events together. So we also hope to have a grafting workshop online, and hopefully the beginning of May with Dr. Hill Craddock down in Chattanooga. We'd like everybody to meet all the staff at some point. I started with Sarah and Jared, our science leadership team, but we also have, our senior management team, which is Betsy Gamber, and we have a new member. His name's Paul Wingenfeld. He joined us at the end of March when all this world was turning upside down, so we are very lucky to have Paul. He took over for Heather Nelson, who had a great run at 5 years being our controller, and Paul came in from the private sector in startup community and startup companies in the healthcare industry, but he is really doing a great job diving in. The first hire I've ever done sight unseen. Just Betsy and I talked to him. We knew he was going to be a great fit, and we're just very, very lucky to have him. So welcome, Paul. So any ideas you all have, we'd be glad to entertain them. Thank you.
All right, we've had a few more questions coming in about the transgenic stuff, but I'm going to put a pause on that because we have a lot of field-related questions and we only have about 15 minutes left. So from Pat Peterson, do you have any pruning tips for us at this time of year? Probably a Sarah question.
Yeah, typically just follow your basic good pruning techniques. We generally don't recommend pruning the research materials, stuff that is still somewhat susceptible to blight. We don't generally recommend pruning them at all. There's not much cause for it. Chinese chestnuts, as long as you follow basic good pruning practices, anything that's promoted for any kind of oaks or other hardwoods, you can follow that. If you're going to prune something, if you must prune something that is susceptible to the blight, even moderately susceptible, we recommend you do that in winter time. Kendra, I know you and your colleagues up there in Vermont have done some winter coring and have seen little to no blight infection after that. So follow good pruning, good sanitation, good pruning practices. Don't go all the way to the trunk. Leave a little bit of the collar. Now's a good time to do resistant or highly resistant material. That's about all I can offer.
Okay. Don wants to know, he said, he just received American rootstock, but with no instructions. He would like to plant today or tomorrow. Any suggestions for the best way to do it?
Anytime you plant bare root stock, you want to dig a hole that is as deep as the root system and twice as wide. That's the best way to get it in the ground. If it's bare root, that's about all you need to do. Don't plant too deep. Don't plant too shallow. Water really well. Don't overwater. One of my favorite quotations by one of our colleagues, Greg Miller, who has an orchard in Eastern Ohio. He says there's two good ways to kill a chestnut, and that's too much water and too little water. So that's really one of the biggest issues that you have in growing chestnuts, especially early on. But as deep as the rootstock, twice as wide as the hole, that's going to be your best bet. The bigger issue with planting any chestnut is site selection. You got to make sure that your site is well drained and has an acidic pH. That's going to be more important for long-term survival and success.
All right. Well, Sarah, sticking with the bare root idea, we have another question. I'm jumping around a little bit, but can an individual or forestry organization or nonprofit acquire bare root chestnut seedlings from TACF for planting and restoration sites? Specifically, I'm located in Western PA and looking to include chestnuts in a strip mine restoration project.
Sometimes we do have various projects that we have online. A lot of our mining projects are done in collaboration with Green Forests Work or ARRI or other mineland reclamation programs. Usually we work together with them to try and coordinate both distribution and projects for that material to go in. So yeah, sometimes it works out, sometimes it doesn't. There are various roadblocks that come along the way for being able to get material out for a lot of it. And Jared mentioned this, like our biggest roadblock right now is just numbers and how many seedlings we have. So we can't guarantee things every year, but we can certainly talk about, opportunities and possibilities and what we potentially can do for future projects.
All right. Steve wants to know, any thoughts on adding the Allegheny chinkapin to the restoration plan?
I want to turn that over to Jared.
Well, all I know about is the work with SUNY ESF, where they have transformed Ozark chinkapin with the OXO gene. And if that gene is deregulated, I imagine that it might be a more simpler, straightforward process for getting the transgenic chinkapin as well through that process. It wouldn't have to go through the entire process that we had to go through for American chestnut. Now, there is an Ozark Chinkapin Foundation, and what they're doing is breeding, and they've located chinkapins and that they have found some resistance. So they are breeding those together. So similar to what the American Chestnut Cooperators Foundation does, where they find resistant trees and breed them together. I think they're doing this similar thing. They're not doing any kind of breeding with Asian species, for example. So there are breeding efforts, and certainly, there's the OXO gene that is a potential in the future.
All right. We have two questions from the same person that are somewhat related, so I'm going to lump those together. Any need for information posted on open source related to TACF or any mention of American chestnut? Any e-learning or gamification efforts?
The Tree Snap app has a kind of gamification, and I was playing the game myself in a way. Like I was trying to win the competition for finding the most American trees, and I can win because I'm paid to do this. And I was in Western North Carolina, where there's you can just go out in the woods and find a chestnut anywhere you go. So I was in the lead for a while, but then some people started taking over and winning. So if you want to go out and find trees and try to beat me or other people in TACF, please do. I encourage you to. So we really need people to find trees, especially in these areas where they're, and I guess what I would like to see in this gamification effort is if we could add more points if you found trees in areas that we are hard to find, like in Ohio or something, like southeastern Ohio, or in Kentucky, way out west side of the state. That those kind of things, they're much harder to find these trees, and they're valuable to us. Like Alabama, Mississippi, those areas where the tree is in the southern edge of its range, and climate is making this less and less suitable for growing American chestnuts. We really want to find trees on that southern edge. So, and they're much more rare. So I would love to see like some kind of weighting for more points, let's say, if you found trees that are in difficult to find areas where the germplasm is rare. That would be amazing. So maybe I'll talk to Tree Snap about that one.
All right. I'm going to stick with some of these field-related questions, but I just wanted to point out to everyone that Linda from ESF did just post some information in the chat about an upcoming webinar that some of their scientists are participating in next week. So if that's something you want to check out, the info's in the chat. Thomas wants to know, does the research include any reference to aspects of the mycorrhizal networks that may be most beneficial to the growth of chestnut trees?
There's been various projects around that. So I know that ESF has looked explicitly at whether or not there are differences in mycorrhizal colonization on transgenic chestnuts versus wild type or hybrids, and found no significant differences in colonization or species. As far as looking at mycorrhizae in restoration populations, there's been quite a bit of work done on that. What the take home for me is that mycorrhizae absolutely help in establishment. They can improve success and take in seed or seedling establishment. They aren't necessary. You can get success in tree plantings without mycorrhizae, and on strip mine plantings, if you don't establish with mycorrhizae, they find that after about 3 to 5 years, you eventually get colonization anyway. So I think it's something that plays an important part and something that absolutely requires more review and study. But that's where we're at currently with that.
All right, I'm going to jump around a little bit because Connor asked a question specifically hoping we could get to him before the end, and we have about 5 minutes, which I imagine, Sarah, you are willing probably to stick around and answer questions, especially the technical ones, or everyone else. But Connor wants to know, how is root rot affecting wild chestnuts in their native range? What about in hybrid orchard trees or transgenic trees?
So the root rot came into the U.S. prior to chestnut blight in the 1800s, early 1800s. And it's thought that it wiped out the trees more in the Piedmont at that time. So there might have been a larger range than what Little recorded, which was the range map that we typically use, that included some of the Piedmont. Although when we plant trees in the Piedmont now, that in areas that do not have the Phytophthora, the trees don't typically do very well. So there's other factors involved, like climate adaptation. The soils are heavy and not well drained, and that impedes chestnut growth. So what are we doing with in the breeding program? We have Phytophthora resistance in some of our breeding lines, and we've been screening all of our chapter material, our material from Meadowview for that resistance. We're developing genomic models that we can actually use the DNA sequences of these trees and predict which parents carry the resistance and which parents don't. And then we screen that material, we inoculate it with Phytophthora cinnamomi in a greenhouse in collaboration with the U.S. Forest Service and Asheville. And this year we're screening about 5,000 seedlings. We expect to get maybe 300 that will live. And then we're planting that material from whatever lives in an orchard in Georgia this year. We're actually using Phytophthora cinnamomi isolates from that exact location, so that when we go out and plant it, we're not moving those Phytophthora isolates around. We're using the same native isolates. So then these trees have resistance to Phytophthora. Our hope is that if we can get the transgenic tree through the regulatory process, then these trees with Phytophthora resistance, which may or may not have blight resistance, we can cross with the OXO pollen and combine resistance to both pathogens.
All right. I just wanted to note, Robert had asked a question about protection. Robert, I answered that in the Q&A and sent you a fact sheet that covers a lot of that. So if that doesn't answer your question after you look through it, please reach out to one of our science staff and we can help you out. Let's see, from Louise, any plans in the works to use aerial photography, satellite photos, drones to spot blooming surviving Americans?
Nothing definitive and nothing that's spelled out. I mean, I know I've had a lot of folks come to us with those ideas. Several people have done drone flyovers. Several people have done airplane flyovers to try and look for trees. There are some roadblocks to implementing that technology. Obviously, it would be great if we could figure out how to do that, but nothing definitive or concrete as to those plans yet.
Okay, on a related note, and then we'll do a quick time check. Sarah said that TACF was interested in knowing the locations of wild American chestnuts. Are trees in national parks or national forests of interest?
Absolutely, and sometimes those are the easiest trees with which to work, especially U.S. forest lands. I will say, even though our biggest push is for the western parts of the range, western Tennessee, western Kentucky, northern Alabama, northern Mississippi, southern Indiana, southern Ohio, those are really the places where we have the least amount of representation currently. We're still interested in knowing the locations and health of other trees. TreeSnap is a great way to document that. And we can download those data and use them for various modeling practices, both habitat suitability modeling and also trying to model where we might find other trees. So the more trees you can find, the better. Flowering trees are our favorite because then we can possibly get nuts from it. But even if we can't get nuts, we are gathering scion wood in the winters and grafting it for germplasm conservation. So yes, please go find trees.
All right, I'm going to break my own rule here and just give you one more related question, also from Louise. Given that climate change is likely to push the chestnut range north, wouldn't it be better to look for survivors at the northern edge of the range?
So that's an interesting question. I think one of the reasons that we're pushing more toward the southern edge of the range is because the southern edge of the range and western edge of the range is where a majority of the diversity is held. And so that's why we're really going after a push to collect that material. The northern edge of the range is actually, we have pretty darn good coverage. Thank you, Kendra, for covering our New England area. So Maine, New Hampshire, Vermont, Massachusetts, Connecticut, those places have really amazing collections of material. I will say northern New York along the Champlain Valley, that's an area where we could use some additional collections. American chestnuts don't typically grow in the Adirondacks. If you find one, that obviously would be an amazing find. But for the most part, we have really, really good representation from the extreme northern parts of the range.
And the other thing around that question is, if the climate's warming, we need to have trees that are tolerant to the warmer temperatures, and that they are in the south, and that's where the diversity is as well. So our push really is like getting all the way through Tennessee. That's like a really important region where almost the entire state is in chestnut range. Alabama, these are the areas where they have the diversity, and the trees themselves are threatened because of the changing climate. So we want to focus on those trees, rescue them first. I saw that, I went to the Canadian Chestnut Council meeting, and they have a small portion of the range in Ontario that there are trees, and they basically know where every single tree is in Ontario, and they're collecting seeds from those trees. They are planting trees nearby if they're isolated. So they call it breaking isolation, where they're able to get a tree with its mate so they can get seeds from them. So there is some work in Canada, and in Canada will probably be suitable. A large part of like Ontario will be a suitable chestnut habitat potentially, with some climate models going in the future. So like around 2080 or something, that big parts of Ontario actually might be ideal because of the soil conditions, well-drained soils. So we will be thinking about assisted migration with some of this modeling that we're doing to understand how chestnut is related, adapted to climate. We might be moving some of this material that we find in the south further north to make it more adapted to the future climates.
All right. So I'm going to ask Katie's question and then lead right into our time check. Katie wants to know if we can have access to the recorded webinar or any future webinars so they can be shared. And with that, we are at 10:34. I'm not sure if some folks have to leave, but Sarah, I'll let you kind of take over on how you guys want to proceed from here. We have probably 8 to 10 more questions.
Okay. Yeah, absolutely. So for most of our webinars, we've been putting them online. So I think once it gets up online, we'll work with Sharon, I believe, who is on the webinar right now, to get that onto the acf.org website and push a link. Because we are planning on having other webinars, I imagine that link will be available in future e-blasts and e-sprouts. So yes, absolutely, it will be available hopefully within the next week or less. As far as a time check, I'm happy to stick around. Lisa and Jared, I don't know what your all's time check is like, but yeah, all right, excellent. Yeah, we're happy to keep asking questions, and everybody's welcome to, take off or join back in as they wish. So thank you all. I will say, if you do have to leave, thank you for coming. I think the most I saw at some point was about 135 participants, which is excellent. I really want to thank you guys for making time and for your interest in the work that we're doing to restore the American chestnut. So thank you guys, and we're going to stick around. Thanks.
All right. Well, we'll keep going with questions. I will say, folks, at this point, unless you have a really, really burning question, maybe hold them until the next one, or shoot one of us an email so that we're not on here until noontime, because I know we could all talk about chestnuts for another 2 hours if we were given the opportunity. So let me get back up here. So question for Jared from Jack. Outside of crossbreeding the transgenic clone with native chestnuts to increase diversity, are there other molecular level genetic or gene splicing strategies that might be used to accelerate diversity?
Well, one of the things we're exploring is, to speed up this breeding process, could we insert the oxalate oxidase gene into multiple different American chestnut trees? Currently, it's inserted into a single founder tree from New York called the Ellis 1 tree. And we would like to ideally insert that same gene into a tree from the south, a tree from the mid-Atlantic, or maybe numerous trees from each of these regions. And then we would basically break that diversity bottleneck that we start with starting a breeding program from a single tree. And one of the things that we've been exploring is you can actually use now CRISPR to insert this gene into the same root, and every tree inserted into the same part of the genome. So that makes, compared to the previous technology where it's randomly inserted into the genome, when you put it into the same place in multiple trees, that means that it will always be in that same location. If these trees, progeny from these different trees cross with one another, they will only have one copy of the gene rather than multiple copies of the gene. If you get multiple copies of the gene, then and they're all highly expressed, then sometimes what happens is the tree just says that's too much and it shuts down the expression of the that gene. So that's what we're trying to avoid. Now, in terms of other genes that we might use besides oxalate oxidase, there has been work with the Forest Health Initiative a number of years ago now to insert genes from Chinese chestnut directly into American chestnut. Genes that look like disease resistance genes that mapped to regions of the genome that are associated with resistance. So we have a number of trials with that material out in the landscape, and we're evaluating how resistant that is. So eventually we could potentially incorporate additional genes besides the oxalate oxidase if we need them to make the trees more resistant. So we're trying to pursue multiple pathways with the biotechnology as well.
Okay, great. Thanks, Jared. I'm going to ask a few questions related, since we got on the transgenic topic, we'll go back to that. But I did want to point out to folks that Sharon has very kindly posted a little bit more information about some of our publications dealing with drone use. In the chat, we had an article in 2018 issue of the magazine, and a photo will be out in the upcoming issue. So just an FYI there for folks. So I did have one person ask, I think maybe if we could just backtrack a little on the real basics. Do we have any information on the latest announcement on the research of chestnut tree modification of adding a wheat gene to prevent the blight?
So that's what we've been talking a lot about with regard to the transgenic American chestnut, and then the public comment period. That all has to do with the wheat gene that's been inserted into the American chestnut for blight tolerance.
Yeah,
right.
So I think Linda posted the executive summary of their petition, and in that petition, there is a lot of research on what are the potential ecological impacts of putting this gene into American chestnut. So they've compared the growth of the trees, wild trees, to this transgenic tree. They've looked at mycorrhizal colonization on roots. They've looked at bee feeding on the pollen, the leaf litter, the tadpoles eating leaf litter. Lots of these experiments they've done comparing the wild trees to the transgenic trees, and they found no significant differences except for the trees. The transgenic tree has enhanced blight resistance. So that is what we're leading with the USDA, is this kind of a lot of experiments to show that this is safe when we release it into the environment.
All right, well, that's going to segue well into the next two questions. So Jim wants to know, with up to 18 months from submission to approval times 3 agencies, does that translate to 4 or 5 years from now for release of the transgenic tree?
We hope not, not necessarily. They're sort of concurrently going on at the same time. The work that's being done now with USDA and the pending public comment period, that's very imminent. And we hope from the beginning of that public comment period to their decision will be 18 to 24 months at the most. EPA is working on their interpretation of the law called FIFRA, which is the Federal Insecticide, Rodenticide, Fungicide Act. It's a 1946 act, and they have ruled that right now they're considering this OXO insertion of the wheat gene to be a pesticide and mitigates the fungus. This is all kind of legal conversation that we've been having. So EPA, although has, as I said before, has been very supportive, they have recommended we submit a registration, go through a registration process, which unfortunately would not be appropriate for restoration because it has to be renewed every year and would constrict our ability to release the tree. But they recommended we do an exemption at the same time. It's a 25B part of the law where we can actually be exempted from that. But that's rulemaking, and that takes some time. So there's been a lot of regulators working very hard on this now. They have huge caseloads. So we're hopeful that the EPA and the USDA will be around the same time frame. There's also a third agency, the FDA, because technically chestnuts are a food, but we understand that's a little less onerous of a process. And again, Bill Powell, Andy Newhouse, and his team have been really great reaching out to all these regulators and developing relationships so that can be a smooth process. We understand that's not going to be quite as intensive as the EPA and the USDA. So we hope it's not going to be 4 to 5 years. We hope it's more like a couple of years because we want to get going on this third prong of the 3-burr process, multiple pathways towards our goal. So that's what my understanding is at this time.
Yeah, the other thing to mention is that we are currently doing some pollinations with permits from the USDA. trees take a long time to grow, as you know. So we are doing these pollinations. We're potentially planting some of these trees out in trials. So SUNY has been doing this for years now already. So we have permits to do this. And if the regulatory process gets through, then we have, by that time, even if it is 4 or 5 years, at least we have trees that are potential parents that we could use for distributing seeds. So we're trying to get ahead of the game a little bit.
All right. Well, Steve wants to know if there is there or have you considered adding a social scientist or scientist to address the cultural, social, and financial significance of the American chestnut to the peoples who lived within its native range?
That's a great question. I don't have a super good answer, but I know there's been work done out there with ethnobotanists and work with indigenous communities. I know Doug Gillis and Steve Barilowitz attended, gosh, probably a couple years now ago, kind of a conference in NC State to talk about the social aspects of chestnut, and especially with the transgenic issue. We're working currently with the Cherokee Nation through Rex Mann, our wonderful spokesman and longtime member of the American Chestnut Foundation, to see how interested they might be in doing a germplasm conservation orchard. We're just trying to have these conversations as part of the whole task force with the transgenic tree. But it's a really culturally significant species. It's just legendary, and I think that's why so many of us are interested in getting it back into the forest. So, Sarah, Jared, I don't know if you have something to add to that.
Yeah, I would highly recommend. We had Jason Delborn from NC State, who spoke at the Pennsylvania, New Jersey chapters webinar on March 21st, along with Jared Westbrook as well, who gave an excellent overview of TACF's current scientific status and projections. That webinar is on the PA New Jersey chapter's website, patacf.org. If you want to see, Jason's a social scientist, and he's investigated a lot of aspects of transgenic American chestnut, not only with indigenous peoples, but also how this is going to impact other cultural or challenge societal norms, and how people are going to be willing to accept transgenic technologies into forest restoration. So if you want to know a little bit more, I highly recommend checking that webinar out, or seeking out maybe some publications that he's put together. And I know one of our attendees, Katie Barnhill, help me, Jerry. Dilling is on here, and she's worked with that as well. And so there's several different social scientists who have worked on this aspect, because it is bigger than just the science. So thanks for that question.
All right, last one on transgenics, I think, unless anything else has rolled in. Has any thought been given to the effects of pleiotropy in regards to GMO?
Yeah, pleiotropy, just for everyone else who doesn't know that term, means that the gene has multiple effects. So it could affect blight resistance, but it could affect something else like growth. It could affect other associations with different fungi. And that is the purpose of the petition process and the research that SUNY ESF has done on this, is to look at, does that gene insertion affect the metabolism of the tree, besides or the growth of the tree? Does that insertion disrupt any native genes in that genome? Does it affect the mycorrhizal associations, all of these things? So yes, that is exactly what that petition tries to lay out in 280 pages. So there's a lot of work done on that already. And so far, you could imagine like 1 million experiments that you could do to evaluate how these trees will do. But all of the experiments thus far have shown no significant differences in terms of these other effects like growth and metabolism and everything, or nutrition, for example, of the nuts. So that's what's in that petition. So I recommend you take a look at that.
All right, thanks, Jared. Getting back to field type questions. I just received my chestnut trees and noticed that all the roots have a 90-degree bend in the taproot. Is that going to be a problem? It looks like they were planted in too shallow of a pot or media. My understanding is the taproot should be straight.
If it's right where the tree meets the soil line and it kind of does one of these and then goes down, should not be an issue. That happens a lot, and I think that is depends on how the nut was oriented when it got planted. The J rooting or the issue comes when that J or turn in the root happens farther down in the root system. Generally, it's not too much of an issue if it's at the top. If it's at the bottom, we'll have to talk. Maybe that's deserving of you sending me some pictures so we can check it out.
All right. James is a chestnut enthusiast in Kentucky who just wants to offer his property if we would like to use it, and says an email response is okay. I don't know if we have access to your email easily, James. So if you want to drop one of us a note, you can find our contact on our website, or who would be the best contact for Kentucky at this point? Probably Tom?
Yeah, it's in Tom's region. So.
Okay. Oh, perfect. So Tom Sayeli would be the contact, and I can put his email in the chat for you. Just drop him a line and let him know what you have to offer, and we can see if we've got a good fit. 2 questions related to tree snap. One was just wondering if you could further explain what tree snap is. And then Olga wants to know if, as we search for and report surviving trees, is there a public log on the tree snap application to avoid redundant reporting? So quick overview, and then if you can address that specific question.
So Tree Snap was or is an application that's been developed by the U.S. Forest Service and the University of Kentucky and various other collaborators. It's an open source piece of software that you can download on your phone. It's available for iPhone or Android, and it was developed to try and catalog and document trees that are under threat. So American chestnut is one of them, but it's also geared toward things like Florida torreya, the white and green ash, white oak now because it's being threatened in this population. So it's a great way to log not just American chestnut, but other species. It will ask you how tall the tree is. You estimate that. How big is the tree? Is it flowering? Does it have the blight? And then the biggest thing that we need are pictures, really good pictures of the leaves and the bark and the blight and things like that. We also ask you to actually send us a leaf sample so we can verify that it's an American chestnut and not a Chinese or something else. Is there a way to avoid replication? Not great, no, but that's actually not too bad of a problem, because one way we can use those data is if we see a lot of people pinging the same tree. One interesting aspect of this is from a citizen scientist aspect, is seeing how are people reporting these trees and their measurements. Are they relatively the same, or are they way off? Like, are some people reporting the diameter is one way or not another? So it's actually kind of fascinating to have this replication of information. Another way is, are there areas where people are pinging a lot of trees versus not? So you can follow really interesting volunteer and citizen scientist patterns. So, I don't want folks out there replicating effort, but, a lot of this can actually add to various aspects of study, not just documenting American chestnuts. Oh, there you go. Perfect.
Lisa's got her tree snap.
Yeah.
All right, just 3 more questions because I know we're running quite a bit over here. Robert wants to know, how effective is mud packing around the injured trunk of an American chestnut tree? Does it really help?
It does. If you catch the blight, so mud packing should only be used for blight. It shouldn't be used for any other injury. If you have a blight canker, the first thing I recommend you do is cut out the canker. You don't have to do that step, but I think that's the most effective. If you can cut out the canker and find the division where the necrotic or black or brown tissue stops and the white or living tissue starts, if you can cut out that canker and get to that margin, you can see how far the blight has gone. Put mud on it, hold up the mud with basically a trash bag and duct tape or some variation of that, leave it on for a year. It will arrest that canker. It should, I won't say will 100% of the time, but it usually arrests that canker It won't keep other cankers from forming. It won't save your tree forever, but it's a great way to prolong the life of a given chestnut tree.
Yeah, and I will say we did have a good article, a good how-to article from the SUNY ESF folks and one of our recent magazines. So if you need instructions, I don't recall which episode, which issue it was, but it was somewhat recent. Frank and Phil are both asking about oak wilt disease. We've seen that at Greg Miller's orchard in Ohio, and just wondering what else we know about it, if we've seen it anywhere else.
I haven't seen it anywhere else. I've seen that folks have done artificial inoculations on American chestnut and Chinese chestnut and chinkapins with oak wilt, and they are susceptible. I have not seen it in the field.
All right, and our last two, we're getting back to transgenic trees one more time. What do we know about the mode of action of the Chinese genes in providing blight resistance? Is it similar to the OXO gene?
Yeah, there's actually a pathway to detoxify the oxalic acid that the fungus produces to create the cankers in Chinese chestnut, but there's other things that are going on in Chinese chestnut. It's like more complex resistance. There is the trees that have resistance tend to form this like woody lignified area surrounding the canker, and that prevents the fungus from growing into the cambium of the tree. So there's a whole set of like reprogramming of cells to form this like physical barrier to the fungus from entering the wood. There's also probably some interesting things that are happening at like a metabolite level, where there's some metabolites that the tree produces that inhibits the fungal growth. And we are actually going to be studying that. We're working with Oak Ridge National Labs where we're inoculating American trees, Chinese trees, and hybrids with the blight fungus. And we'll be looking at which metabolites get put out by the tree's wood when they're infected compared to the Americans and the Chinese and the hybrids. So there's still a lot that's not really known about how the Chinese tree resists blight. We know it's complex for sure. There's many genes in the genome that are affecting it, but we don't actually know, like, besides the oxalic acid detoxification and besides this like woody tissue formation, what other mechanisms might be involved. It's an area of active research right now.
All right, last question, which I think is more of a comment, but I'm going to read it and let you guys decide if you want to discuss a little bit further. The social scientist question came from the perspective of using that research to support in DC the restoration applications. Approval of the transgenic tree is the best chance of restoring cultural benefits and significance to the Appalachian population. I imagine that having a pool of legislators from the Appalachian region has the potential to grease the application and approval process. In other words, what's the significance benefit to their constituents?
Well, that's an area that we haven't really had the capacity to tackle, is the legislative side of things as much. We did get, did some outreach. They contacted our office from Ohio. Senator Sherrod Brown is a chestnut restoration enthusiast. Brian McCarthy, Carolyn Kiefer, and Leela Pinchot met with them, and Barb Tormelin. So we're trying to, have both sides of the aisle, because we're strictly apolitical and nonpartisan, to, embrace this hopeful mission, because it could bring jobs eventually if we do large scale restorations down the road. So I have met with Senator Brown's aides a couple of times, and then the most recent one, that those 4 wonderful volunteers, leaders, met in Columbus. So if you all have, you think you might have an interested legislator or someone in government that would help us. I know the New York chapter has been able to get some funding through their New York state folks. So I think there are possibilities out there, and we're wide open to do that. We don't have a government relations department. I'll put it that way. We're pretty lean and mean. So we've been able to get to DC a number of times, but it's a great comment. I think it could really raise the consciousness of our political spectrum if we did something that was optimistic. You know, it's such a divided country, and this could bring people together, hopefully. Good comment. Thank you.
All right, we had one final question come in. Everyone else, I appreciate them, but please hold on. Hold them at this point. We're almost at 11. Are we working with the ACCF on straight Americans?
We actually have some of these same large surviving American chestnut trees that ACCF has used in their breeding program. We also have progeny of those trees that we've crossed them together. And what we're doing, and they actually do have some resistance. It's amazing. And I'm very curious about why these trees are resistant. We've been able to screen progeny from them. We can confirm that their kids also have resistance. So that means that there's a genetic mechanism involved. And so what we're doing is we're sequencing the genomes of all of these large surviving trees. Like there's a tree in Pennsylvania called the Orks tree. There's a champion tree in North Carolina, numerous trees in Virginia that we know about. So we're sequencing their whole genomes, and we're comparing their genomes to the genomes of susceptible trees. And we're hoping to see that there's some kind of systematic differences. We need to figure out what those differences are and what genes and mechanisms might be involved. But we are also, I mentioned earlier about doing metabolite profiling of trees. And so we have one of those large surviving trees that we're comparing to a susceptible tree, the tree that we actually have a genome sequence on. So we're trying to look at what's happening down at the molecular level as to why these trees have resistance. But they certainly do. It's not like it's moderate resistance. They still get the blight. They still have cankers all up and down the stem. But it's amazing that they survive.
I do want to add one thing. If you guys are in South Central Pennsylvania when things open up, if you've ever been to Cadora State Park, Hanover, Gettysburg area, near the campground, there's a sort of a large surviving American orchard called the Kelly Amherst Orchard. That's one of the best ones that I know of, the easiest accessible orchard to get to, that has wild type American chestnuts displaying native resistance. So if you want to see what intermediate resistance looks like in the native population, that's a great place to do it. Is that it, Kendra?
That is it for questions.
All right.
And thanks for all the great questions, everyone.
Okay, fantastic. Well, we covered a lot of territory. We really appreciate you guys joining us and being interested in the work that we do. So we're putting together something for next week. You guys should get some information about that relatively soon. And thank you again. Keep up the good work that we know you all are doing toward this great effort. So, right.
Yes, thanks everyone. We really appreciate your engagement. This really helps keep us motivated and ready to get on the other side of this. If you can, a couple other call of actions. If you can, haven't talked to a friend in a while that you haven't reached out to, now that we're at the comfort of our home, you can reach out and maybe recruit them as a new member. We really depend on our membership. 95% of the revenue of our work comes from private philanthropy, members, donors, foundations. We get very little funding. We get some funding from the U.S. Forest Service. They're our main partner there, but help us recruit new folks to tell the story, because it really is a hopeful story that we all want to share more widely. And when we come out of the other side of this, let us know if you have speaking gigs that you can tee up for us. We've done a lot last year. Sarah and I just got back from Florida in the nick of time from a Jupiter Island Garden Club down there in Southeast Florida. It was really a great group of ladies, and Kendra and I went to Beacon Hill Garden Club. So the Garden Club system is really important to us, and they are very influential. They can help us out in DC, to one of your points. And so those are a couple things you can do, and keep an eye out for the public comment period. We will definitely be sending materials out on that. And I just wanted to add my thanks to Sarah, as you all are wonderful volunteer leaders and members. So keep up the good work, and we'll see you on the other side of this.